Each organism is adapted to its environment. The form of nutrition differs depending on the type and availability of food material as well as how it is obtained by the organism. The organisms, those cannot synthesize their own food but depends on other plants and animals are called heterotrophs. All animals and non-green plants like fungi come under this category. Depending upon the mode of living and mode of intake of food, heterotrophs may be parasitic, saprophytic, or holozoic. Parasitic. Parasitic organisms or parasites live on or inside other living organisms called host and obtain their food from them. The host does not get any benefit from the parasites. Example, cascota, then hookworms, leech, tapeworm, etc. Saprophytic organisms or saprophytes derive their food from dead organisms. They secrete enzymes that are released on food material outside their body. These enzymes break down the complex food into simple forms and they are absorbing it. Common examples are fungi, mold, mushrooms, yeast, etc. In holozoic nutrition, complex organic substances are ingested, that means taken, without their being decomposed or degraded. After intake, such food is digested by enzymes produced within the organism. Digested food is absorbed into the body and the undigested food is expelled from the body. This kind of nutrition is found mainly in non-parasitic animals, simple ones like amoeba and complex ones like human beings. Different organisms obtain food in different ways. Nutrition in unicellular organisms like amoeba involves ingestion by the cell surface. Amoeba takes in complex organic matter as food. Amoeba first identifies its food. It then throws out small pseudopodia, projection of cytoplasm, also called false feet. These pseudopodia enclose the food particle and prevent it from escaping. The food enclosed in a cell membrane forms food vacuole. The complex food is broken down into simpler molecules with the help of digestive enzymes. The digested food is absorbed in the cytoplasm and undigested food is expelled out through the cell membrane. In paramecium, a unicellular organism with a specific shape, food is ingested through a small special opening, that is cell mouth or cytostome. Food is brought to this opening by the lashing movement of cilia that cover the entire surface of the body. Next, we can study nutrition in human beings. The alimentary canal and the glands associated with the digestion constitute the human digestive system. The alimentary canal in human beings extend from the mouth to the anus. The mouth consists of oral cavity through which food is ingested. It is bounded by lips and cheeks and contains gums, teeth, a tongue and muscles. The tongue tastes food and moves it into pharynx. Teeth help in biting, cutting and chewing food. Teeth masticate the food and this makes it easier to swallow food and increases its surface area for various digestive secretions to act on. The four types of teeth are incisors, canines, premolars, and molars. Our teeth are covered with a hard protective covering of enamel. The enamel covers the dentine, which is a yellowish substance forming the bulk of the tooth. When we eat sweets, chocolates, and ice creams, bacteria act on sugars and produce acids which soften the protective covering. The bacterial cells and food particles stick to the teeth and form dental plaque. If the teeth are not brushed properly after meals, bacteria may invade deeper into the teeth. This leads to infection and a toothache. The presence of food in the mouth stimulates three pairs of salivary glands to secrete saliva. Saliva has salivary amylase, a digestive enzyme that breaks down starch into maltose. Maltose is a simple sugar. The oral cavity opens into the pharynx. The swallowing mechanism guides the masticated food through the pharynx into a tube called esophagus. Esophagus is a muscular tubular part of the alimentary canal. The muscular walls of the esophagus moves in a rhythmic wave-like manner which carries the food down to the stomach. This muscular movement is called peristalsis. From the mouth, food is taken to the stomach through esophagus. The stomach is a large organ which expands when food enters it. The muscular walls of the stomach help in mixing the food thoroughly with the more digestive juices. The stomach has an anterior cardiac and posterior pyloric part. The wall of the stomach contains gastric glands which secrete gastric juice. The gastric juice contains hydrochloric acid, mucus, and enzyme pepsin. The hydrochloric acid in gastric juice converts propepsin to active pepsin and also kills bacteria. The mucus protects the stomach lining and glands from being digested by gastric juice. About 3 liters of gastric juice is produced per day. Excess secretion of gastric juice, particularly in an empty stomach, erodes the inner lining of the stomach. This erosion causes lesions or round depressions called a peptic ulcer in the stomach walls. Digestion of protein begins in the stomach. Pepsin, the enzyme, breaks down proteins into peptones. The exit of food from the stomach is regulated by a sphincter muscle which releases it in small amounts into the small intestine. From the stomach, the food now enters the small intestine. This is the longest part of alimentary canal which is fitted into a compact space because of extensive coiling. The length of the small intestine differs in various animals depending on the food they eat. Herbivores eating grass need a longer small intestine to allow the cellulose to be digested. Meat is easier to digest, hence carnivores like tigers have a shorter small intestine. The small intestine is the site of complete digestion of carbohydrates, proteins, and fats. It receives the secretions of the liver and pancreas for this purpose. Liver is the largest gland of the body. It performs many functions. It secretes bile, which helps in digestion. Bile juice produced by the liver is stored in the gallbladder. There are two main functions of bile. First one is it emulsifies fats by rendering them soluble and breaking them into small globules. In this form, fats are better exposed to the action of the enzymes. Another function, the acidic food coming from the stomach becomes alkaline when it is mixed with the bile. This is important as the intestinal enzymes catalyze the breakdown of food only in an alkaline medium. The pancreas secretes pancreatic juice which contain enzymes like trypsin for digesting proteins and lipids for breaking down emulsified fats. The walls of the small intestine contain glands called intestinal glands which secrete intestinal juice. The enzymes present in it finally convert the proteins into amino acids, complex carbohydrates into glucose, and fats into fatty acids and glycerol. The digested food is taken up by the walls of the small intestine. The inner lining of the small intestine has numerous finger-like projections called villi, which increases the surface area for absorption. The villi are richly supplied with blood vessels which take the absorbed food to each and every cell of the body. Then it is utilized for obtaining energy, building up new tissues, and the repair of old tissues. The unabsorbed food is sent into large intestine where more villi absorb water from this material. The rest of the material is removed from the body via the anus. The exit of this waste material is regulated by anal sphincter. Next, an activity to demonstrate the action of saliva on starch. Materials required are 2 test tubes, iodine solution, starch solution, and saliva. Take 2 test tubes, we can name it as A and B. Add equal quantity of starch solution in both the test tubes. Then add saliva to the test tube A but not in the test tube B. Leaves both the test tubes undisturbed for 20 to 30 minutes. Now add a few drops of dilute iodine solution to the test tubes A and B. We can see that the test tube A does not show any color, but test tube B shows blue color appearance. Test tube A has no starch, while in test tube B starch is present. That shows blue color after iodine test. Action of saliva on starch is clearly visible in the test tube A. Starch hydrolyzes due to the presence of saliva. Saliva contain an enzyme salivary amylase. This salivary amylase breaks the starch into soluble sugar. So the test tube B only appears blue due to the presence of starch.